So we're going to talk about the alternative complement activation pathway. So basically what the complement system is just 30 serum proteins and membrane components that are involved in the function of lysing cells. They also are involved in the inflammatory response, and they clear immune complexes as well as promote phagocytosis. So they're really important, and they're pretty complicated, these different pathways that they have. So I'm going to try and break it down here as simple as possible. So one of the pathways is the alternative complement pathway, which I'll talk about right now. So there are different proteins that make up this complement pathway, but C3 is the most abundant floating around in the liver. So what happens is there's spontaneous cleavage of C3. So let's see our pair of scissors right here. Randomly cut C3, and then what that forms is C3A and C3B. C3B is what we're what we're interested in. So after C3B is formed from the cleavage of C3A, it's very reactive, and it combined to either one of two common chemical groups, so amino or hydroxyl groups. And since many proteins and carbohydrates that make up the surfaces of invaders, like bacteria, have these amino or hydroxyl groups, there's lots of target cells for this bacteria to bind to. So let's picture a little bacteria cell right here. Here's our bacteria cell. C3b here is going to recognize it, and it's going to hook on to this bacteria cell right here. So once that has happened, or if that doesn't happen, sorry, I should mention this as well. If this doesn't happen, there's nothing for it to find. It has about 60 seconds to find a binding partner before being degraded by water. Okay, so that's important. That is one of the regulatory factors of the complement system. So once C3b is stabilized to this bacterial wall, another complement protein, complement B, will come in. So I'm going to redraw this down here. Let's make this bigger. So we have this. We have the C3b here. And then another protein called B will come in and join in. Okay? After that, a protein D will come in and cut the B so that you eventually form C3BBB. This is also known as C3 convertase. After this happens, you have kind of like a chainsaw methods here. So let's draw this out. So this C3b molecule is, here you got the C3b, and then you have this, do this in red, you have this little like pointy sharp thing that is your B that has just been cleaved by factor D. So factor D is very important for this. And this is still bound to the bacterium right here. So it's still bound to this bacteria. And then what happens is you have this kind of negative feedback loop where C3 convertase can actually cleave a bunch of C3 molecules to yield more C3 convertase. And that just happens over and over and over and over again. What eventually happens is C3b can actually bind to another molecule, or C3BBB, or C3 convertase, can bind to another molecule of C3b. So C3b will come in and join this. And then what this is called C5 convertase. Now, what is C5 convertase? It can cleave C5 into two pieces. So C5 gets cleaved into two pieces, one of which will be C5b. Now, C5b is really important for the formation of the membrane attack complex, or the MAC attack complex. So this forms part of the MAC attack complex. Okay, so what is the MAC attack complex? It is a series of proteins. So once again, we have our bacteria cell here, and then our membrane attack complex is starting to form here. So in this little kind of pole, we have C5b, C6, C7, and C8. Now this is kind of a latent form of the MAC attack complex. What it needs is C9 in abundant value to come in and form a pore that allows for the leakage of all of the bacterial or components, anything that's inside the bacteria to leak out, and then that bacteria is toast. So that is what the alternative complement activation leads to. So we have C3b, which is floating around in the blood and the tissues, made in the liver, which gets spontaneously cleaved into C3a and C3b. Well, we're interested in C3b. It looks for a binding partner. It has 60 seconds to do it. So think of it like a sticky grenade, and if it doesn't do it in 60 seconds, it's neutralized by water. Then you have B combining with the bound C3b on the bacterial membrane right there. After that, factor D comes in and cleaves B to form C3BBB, or C3 convertase. C3 convertase then has a positive feedback loop, so it just continuously makes more C3 convertase by cleaving C3 molecules to form C3b molecules. Okay? Eventually, C3b will combine with a C3 convertase to cleave C5 into C5b and C5a. C5b is what we're interested in because it is involved in formation of the MAC attack complex. So the MAC attack complex obviously attacks cell membranes, and then you have cell death. So in the next video, I'm going to talk about the lectin activation pathway and then the classical activation pathway. So this obviously is involved in the innate immune response because no antibodies are needed.